Hello. Today we are going to speak about prenatal development of the human nervous system. This lecture is part of 550 Developmental Neurobiology, which is part of the Graduate Certificate for Educational Neuroscience Applications that is offered by Bridgewater State University by the Center of Educational Neuroscience Applications. So first, looking at the entire nervous system, the human nervous system, and how it is formed. First, you need to think about how a human being, for example, is designed. Obviously, we are animals. Animals means to move around, animation, and you need a brain in order to be the central control system. And from that brain, you bring in down connections or cables, and out of these cables, you get out nerves, and then you use these nerves to innervate different parts of the body to control it, and also to enable the entire body to live and move and survive. So the whole purpose looks like connecting some kind of structure, whether that's a building or the electric, grid inside of a robot, or some kind of animated, one of these animated things. So we are going to look at the basic function and the basic structures and the basic functions that give rise to the human nervous system. But at some point, we'll speak about the structure of the neuron itself and the neuroglia, because we are going to be speaking about different terminology afterwards, and we want to have something to connect that information with, so that we can with the understanding of the neurons and how they connect and how they are supported, then we are able to see them in a larger kind of picture. The formation of the nervous system is determined by DNA. So initially, it's like a seed. The seed, say an apple seed, it has the DNA, it has the sequences, and it has the programming that enables it, as soon as water, as soon as that seed is in the right soil, and as soon as there are conditions like water and other conditions in the environment, then the DNA is going to start to express itself. And it follows a sequence that is extremely precise, and it continues from that point. So we have genetically predetermined structures and a sequence. However, the effect of the environment is going to cause alterations to that genetic map or to the genetic programming that was already predetermined. So we'll come back at this point later in the lecture. The process of how genetics and epigenetics interact together in order to sculpt or to kind of design the human system in a way as to make it the best fit to the environment it finds itself in. That process and the interaction makes us think about the nature and the nurture kind of discussion. It is not really a debate. In the past, it was raised, and it is an argument that shouldn't have taken place at all. With our understanding today of the brain and the nervous system and how the interaction of the environment goes. With the brain, we understand that it is like having, say, a bill, some kind of, say $1. If that dollar is split in half, and you have one piece in each, one piece in each hand, you are not going to be able to buy anything. You are not going to be able to do anything with that half of the dollar in your hand. And the other half is the same. Either piece is not going to be useful. In order for that to function, and fulfill the purpose for which it was designed, that dollar bill, it has to be one piece. So in the past, they tended to take one thing and they kind of mutually exclusive. If this is more powerful, then that is not as important. No, both of them are needed. You cannot just have an environment and have, if you want to like using a computer, first you have to have a computer. And when you get the computer, how you use it and how you maintain and all of this, that is going to determine the efficiency, the use of the computer, and so on. So that argument of the nature-nurture debate it should not be a debate in the first place. It should be that we have a genetically predetermined structure, construction, and it is left in draft form until the baby comes to, when the baby comes to life, when the baby is born. And from that draft form, the environment can create, conditions to alter that and to modify the body and the nervous system and everything in order to make the organism, to make the human being the best fit for that environment. So it's a survival mechanism. So the whole nervous system has two major categories of cells, neurons and glial cells. The word glia comes from glue. So glia means glue in Latin. So you have a neuron, and then you have all these cushioning, cells cushioning it from all sides. And of course, that's how they saw that in the past, and they just look at these structures that, these are like glue holding axons and holding neurons in place. Of course, they are more important than that. But glial cells outnumber, in some cases, if every neuron, you might have about 50 glial cells. So they outnumber neurons. There are, in the cerebral cortex itself, there are more than 25 types of neurons. And we'll just, touch upon just a couple of them. This is not something that we particularly are going to focus too much on, but just to give you an idea about the diversity of brain functions as based on brain structures. And then, so glial cells perform different functions. First, I'll just touch upon maybe a few of these functions. Number one, they form one kind called the astrocyte on the left here. Astrocyte. Astro means star, and cyte means cell. So astrocyte means the cell that is shaped like a star. You could see this cell is responsible for forming the brain-blood barrier. It has extensions, and it sends, at the end of each extension, it has like a pod. Pod means foot, podiatrist, foot specialist, so foot. And it anchors, it kind of connects onto a blood vessel on one side, several pods, and on the other side, it is going to connect onto a neuron. So what it will do is, the inside of the blood-brain barrier, there are specialized gates, channels, that are going to take in glucose and the nutrients that are needed by the neurons so that they can survive. And then that the nutrient and everything else is basically shut out. Water, can go in and out, freely. And then all of that is processed in the body of the astrocyte and then shipped to the neurons so that the neurons will get, the nutrition. So that is one kind. And you could imagine the neurons cannot survive without these. Another kind, two kinds. One is called oligodendrocyte. Oligodendrocyte is named after a particular sugar molecule that that is prominent in the formation of these cells. And this is the cell body of the oligodendrocyte. And you could see that it has several arms, several extensions out of its body, and every arm has at the end of it a kind of a segment of myelin. So that myelin wraps itself like a jelly roll about 3 or 4 times around a piece of one axon of, maybe every arm can insulate a few pieces of the same axon, but also other arms are able to extend and hold on and insulate different pieces of different axons that belong to different neurons. So you can imagine if there are, say, 10 axons coming down, and I am an astrocyte, so I am gonna stand and extend. I have 10 arms or more, maybe 20 or whatever, and I extend and I hold and hold, all these neurons as they are around me and hold them. So in addition to forming the insulation for the axons and making the signal pass effectively and efficiently and preserve the energy, in addition to that, I am also anchoring these axons and holding them in place. So I am also providing structural support. Okay, another kind of, the oligodendrocytes form myelin in the central nervous system inside of the brain and the spinal cord. The other kind, this is a motor neuron, and you could see on the same motor neuron, separate segments of insulation of myelin. Every single segment of these is one cell by itself. It is called the Schwann cell. So Schwann cells, the Schwann cell kind of buds or kind of grows like a little piece of a structure, like, it gets curved at the beginning, and then it wraps itself around the axon, and it continues to wrap itself and wrap itself and wrap itself. It goes up to 16 layers. And depending on the number of layers that you have, that is going to determine determine the functions of that the person is able to perform. Like a very young child is not going to have so many layers of Schwann cells on axons. So every single segment of one motor system or sensory peripheral nervous system axon, every single axon is going to have multiple segments of Schwann cells. So Schwann cells myelinate the axons of peripheral nervous system neurons. So that's the third kind. Another kind that is a glial cell, it's called the it's part of the immune system, and it performs it's like white blood cells, but these are modified and smaller in size a little bit, and they are called macrophages. So, but in the inside of the brain, they particularly maintain the function. They maintain that's kind of say, for example, the immunology or immunity inside of the nervous system. And when there's debris, when there are infections and so on, they just move that debris, they clean up, they maintain, and they kill pathogens and prevent them from invading the brain and the spinal cord. So another kind called radial glia or radial glial cells, and that is each cell is shaped like a little cylinder, small piece, like think about pieces of like macaroni, and then they get together and connect to make longer and longer and longer tubes, and then they extend from the site where the cells are born to the very end, to the site where cells are supposed to migrate. And then as the cell migrates, as soon as it is born, it begins to migrate. While it is migrating, it holds onto that rope or that hose that's created by the radial glia. And then when it reaches its destination, that neuron is now mature and is able to perform immediately when it lands in the site where it belongs. There are many other functions, numerous functions of glial cells, but this is just to give you an idea why glial cells are critical. And for example, when there's degradation of the myelination, the person will have motor disease like ALS, like Huntington's disease, all of these neurodegenerative diseases that are motor in nature, and then so they result from demyelination, degradation of the myelin sheath. So without this, the person cannot survive. In terms of the neurons themselves, they come in different shapes, they come in different forms, and these are some of the basic structures. There are some like motor neurons, sensory neurons, there are pyramidal neurons, Purkinje, or these exist in the cerebellum, for example, the biggest cells in the cerebellum. And then you have some that are bipolar in relation to where the body is and where the axon and the dendria are and so on. Again, we're not going to get too deep into these details, but just to tell you that there are many kinds of neurons based on structure and based on function. Now let's look at the basic structure of the neuron. What makes the neurons special is that they are able to communicate through signals by firing electrical signals or by using chemical or electric synaptic transmission. And we'll speak about these two types afterwards. So the motor neuron is a simple kind of neuron that shares basic structures and basic, kind of organs, with the other kinds of neurons. So it is used commonly to demonstrate and to show the basic structure of the neuron. So a neuron will have a cell body. That cell body has a nucleus, and the nucleus, of course, has the DNA, and it also has organelles that produce little structures to maintain the cell and to copy the DNA and so on. So the cell body, soma. And then out of the cell body, you have extensions that we know as dendrites or dendria. These inside are continuous with they are continuous with the body of the neuron, and they also have organelles inside. They have places to manufacture products that are needed for the neurons and especially for the dendrites themselves. And there are thousands and hundreds and thousands of them on one cell. Even the dendrite itself, it has important structures called, like little buds, called dendritic spines. These dendritic spines are the sites for connections with other neurons. What I wanted to say here is the dendrites are the primary target for connecting with other neurons. So a neuron receives signals through dendrites, and the neuron sends signals to other neurons. It sends signals through its axon terminals. So here's the cell body, dendrites. They receive signals from other from the axon terminals of other neurons. And then we have this little triangle that connects the axon with the cell body. That little triangle is called the axon hillock. The axon hillock is going to determine if this particular cell is going to fire or if it will not, or if it will, inhibit. Let's say, for example, that there is an axon terminal like this of another axon, of another neuron, comes in and it connects with this dendrite of this neuron. The axon that is sending the signal down is called the presynaptic axon, or the presynaptic neuron. And then this neuron that is receiving is called the postsynaptic neuron, because the synapse is the point, and what is behind the synapse is the one that receives. What is in front of the synapse is the one that gives the signal. So now, the signal, this say this neuron, let's say it is one of the big neurons. Some of the neurons can get signals or can connect with more than 10,000 neurons. So every one of these 10,000 neurons is sending a signal telling this or giving information to this neuron, either to say fire or either to say don't fire, and so on. So signals can be inhibitory or excitatory. So all of these signals, let's say 10,000 or more or less, sometimes 1,000, sometimes less, and so on. All of these signals, whatever they are, find their way after they are processed and so on. They go finally to the axon hillock. And now the axon hillock, it has the ability to fire, that signal that has come in so that it can reach other neurons on the other end. However, the process requires a lot of processing. So the axon hillock is going to look at the signals that are coming in, and it will determine by the majority of the signals that are coming in if it will fire or if it will inhibit. If the majority of these signals tell it to fire, then it will fire a signal. The signal, after the signal that is received, but it is modified in many ways, that signal is going to be fired by the axon hillock. And that signal is going to travel through the axon. As it goes through the axon, then it reaches the axon terminals. As you notice the arrow, the signal goes in a single direction. It is not a bidirectional. It is a unidirectional pathway. The signal cannot go back, and it just goes into one direction. So once it reaches the synaptic terminals here at the end, these synaptic terminals are connected to or are synapsing with dendrites of other neurons. Sometimes they also synapse with the body of other neurons or even with axons of other neurons. But the idea is these synaptic terminals pass on copies of that signal to all these neurons that are connected to it on the other end. So we're going to look at the structure later of the synapse and how, it is formed there, how the signal, when it reaches the synapse, we come to this end, but we'll look and kind of magnify this point and say what goes on here when we speak about basic transmission. So you could see here in this image, how neurons are connecting with other neurons. Sometimes the axons of a neuron connect right on the body. Sometimes they connect with dendrites, and this is the greatest number of neurons connect to dendrites. And then some of them, connect onto the axon itself. This is not demonstrated here, but they can just get their axon and connect on this, axon itself of another cell. So the dendrites themselves, they are lined up with receptor channels so that they can communicate with the other cells, and they can receive signals. I explained that how the axon has myelin and how that myelin is formed, and the axon hillock here, and how it is responsible for processing signals and firing them to other neurons that are part of the circuit or that are connected with it. Then, before I speak about the synaptic structure itself, I wanted to mention that there are two kinds of synapses. A synapse is the point where one neuron communicates with another. You have an area, the neuron that is receiving a signal, like the postsynaptic neuron, has an area that's kind of curved in like this, and then you have the synaptic terminal ending with some kind of foot called pod, and they kind of come close together. There's a little gap between them. They do not quite touch, but there's a little gap between them. And there's space, that gap is called the synaptic gap or the synaptic cleft. And in that gap, they exchange, the neurotransmitters flow, and then they dock onto the receptors. So the cell or the neuron that receives the signal along the membrane is going to have many of these protein channels that only allow certain things to go from the extracellular space to the inside of the cell. So we have two kinds of synaptic synapses. One is called the electrical synapse, and one is called the chemical synapse. The majority of synapses in the human body are chemical, where you have a neurotransmitter and it does its work and so on. The electrical synapse, I'm going to show you, and that there's no gap in between the membranes of the two cells, two neurons. They're kind of connected with channels, directly connected. So whatever signal you have in one cell, you could just shoot it directly, and it goes through the channel without having to release neurotransmitters or anything. This is a fast-acting, powerful way of neural transmission that is used, for example, by the squid or by different animals, but it's not the dominant one in the human being. We have some of them, but they are not the dominant kind. So we use the chemical synapse. Chemical involves releasing some kind of neurotransmitter or neuromodulator. When it is released, it is going to kind of bind to the receptor channels of the neuron that is receiving signals. So this, if you take a piece of the dendrite here, for example, magnify it, and take a piece of the axon that is, the axon like this, that's connecting. So piece of this axon, for example, and a piece of a dendrite. So here, this is that piece of the axon terminal, and the piece of the dendrite, and it is curved in a way so that it will kind of fit well with the pod of the synaptic terminal. And in between, there's a cleft, that little tiny gap that is between them. There are different adhesion molecules, and one kind of them is called synapsin 1. There are different categories of synapsin. These are like proteins that hold the two pieces together. They hold the synaptic branch and almost like Velcro, and the membrane of the postsynaptic cell, and they hold them kind of together. And the more you are exposed to the information, the more you practice and so on, the stronger these synapsin connections will be, so that you are not going to forget the information quickly. Once a synapse separates, the information on it is gone. So as long as it is connecting, then the information, you can access it. So let's look. The electric signal comes from the axon hillock above, comes down until it reaches the axon. Once it reaches, it induces formation of little vesicles like bubbles, and these vesicles are going to have inside of them neurotransmitters, the neurotransmitter that is needed for this particular, action. And these neurotransmitters then, in the vesicles, when the electric charge, the firing, is coming down to this area, it is going to also drive these vesicles closer to the walls of the membrane, the membrane of the synaptic terminal. Once it reaches there, it touches, little hooks called synaptic snares come out and just clutch onto it and then pull it to make it fuse with the membrane. Your goal here is to release the neurotransmitters outside of the cell membrane without allowing anything to come in. You want to control the inside of the cell. So by fusing part of, then finally it will fuse that vesicle, that membrane, with the membrane of the synaptic terminal, and then this way effectively it releases all these neurotransmitters outside without having to open itself to the environment. So then the neurotransmitters are going to dock onto these receptor cells, and then they can either open them to have chemicals like sodium, potassium, and so on go in and out, or to inhibit the function of the cell to close the channel. So as soon as the signal then, as soon as the gate is opened and these chemicals go in, then the neurotransmitter does its work. Then the neurotransmitter takes off and gets recycled or disassembled in different ways. Some of them get recycled, some get disassembled, some get stored until they are needed again, and so on. And then they are sent back into reuptake gates or channels of the synaptic terminal that already sent them. And then they will, as they go in, they wait to be again recycled, reused again, and put again in these vesicles and kind of used back and forth again. Some of the neurotransmitters do not last much, and they could be disassembled and just discarded and totally. And some could be if there's more, they could be taken, absorbed by some glial cells like the astrocytes, and they will be taken and shipped to another neuron that may need them. So this is the construction. This is a protein channel, and the idea again of it is to control what goes into the cell, what goes out. And here is this, you can see magnified. In the bottom inside, there are mechanisms and gates that will enable the cell to close and open as well. And some receptor channels allow only calcium, some potassium, so they are specialized in different ways. But it's important to know that the neurotransmitter itself does not go into the postsynaptic membrane. It just does its work from outside. It functions like a key into a lock. So it has its own site, and once it goes in and activates this particular receptor that is made for it, then it does its work and then takes off. There are more than 100 neurotransmitters, neuromodulators that act on cells like these to enable the flow of chemicals like sodium, potassium, chloride, and so on onto the cell and out. And there are two different kinds, but we don't want to go too much into these technical details. And I just wanted to give you a basic idea. There is a kind of receptor that doesn't open gates. Yes, it takes a neurotransmitter, but it doesn't open and close gates. What it does is it activates a second messenger inside of the cell to bring the signal, to broadcast the signal inside of the cell. So now let's look at the development of the nervous system before birth, from the point of conception to the point of birth. First, with conception, when fertilization occurs, very, very soon, in 72 days or so, 72 hours or so, you are going to have, like one complete cell that has one layer covering, the kind of lining it up completely, and it is going to start, of course, to be dividing very, very rapidly. By day 13 of conception, or the beginning of the third week of conception, the cell now has 3 layers. It has, the egg, the egg. It has now divided so many times, and it makes 3 layers. These 3 layers are called the gastrula. And so you could see here where it goes from 1 layer and it starts to, form 2 layers, and eventually it's going to have 3 layers of cells. So the first, from inside, you have the first is the endo. We're taking it from outside. I'm looking for the endoderm. So the endoderm, the inner layer, and then the mesoderm is the middle layer, and then the ectoderm is the outside layer. So the endoderm develops into the digestive tract and the lungs, pancreas, and the liver. And then the mesoderm, meso means middle, the middle layer develops into bones, muscle tissue, blood, and a big part of the vascular system develops out of it. And then the ectoderm is going to develop into two things, the skin and the nervous system. Just to make you appreciate that the skin is totally covered with nerve endings to enable us to feel and to sense anything that's around us. So at this point in time, when there are three layers of cells, on top of the cell, let's go back here. On top of the cell, you're going to have a groove, like some kind of a plate is going to develop on back. So here, this is another view, like from the top here, and you could see there's a furrow in the middle, like here, and it goes from front to the back of the cell. The cell gradually elongates. I keep calling it the embryo. Let's call it the embryo, as now it is. So you start to have some kind of a flat, a little margin that is flat, and it goes from the beginning to the very end of the embryo, the back of the embryo. And then it goes downward, it curves down to form that furrow or the neural groove, and it extends from front to back. And gradually the edges of the furrow kind of elevate and keep coming closer, and then they fuse like this. Once they fuse in the middle. As you could see, now they're beginning to fuse, and here they fuse totally. This way, that groove, now as it has fused, it becomes a cylinder. It becomes a tube. So the neural groove, when it fuses again, it forms a tube, a cylindrical tube that goes from front to back, and that is the neural tube, inside of which cells are born and out of which cells migrate, neural cells. And you can see in the video, the animations, and will show you clearly how that forms. Now, all of this, at this point, around day 25, between days 23 and 26, then you start the fusion that I mentioned, when the neural groove top edges start to approximate, and then they begin to fuse. They fuse at the middle, in the center. And this center is close to about the brainstem area, final lemmitube. And then from there, when it closes, the fusion goes into like a zipper action, kind of, as you close a zipper, goes anteriorly to the top segment. The top segment is going to continue to close until it reaches the top. The brain is going to be here. These two pieces here will be the two sides of the brain. And then the other side, posterior or caudal area, is going to continue to fuse back, and that is the spinal cord. You could see it now normally, fused. And now if the fusion of the anterior segment doesn't occur, then the child will have, the fetus will develop what is known as anencephaly. And that means that the brain is not going to develop to the end, and the skull itself is not going to develop. And these babies, they don't live much after they are born. And if fusion in the anterior and the posterior area doesn't go as planned or typically, then that is going to cause spina bifida, which means that little areas of the spinal cord will protrude out, will be basically out of the spinal column. And that causes, paralysis. And even if you are able to correct it after birth, that problem doesn't reverse the damage that happens, unfortunately. Both conditions can occur as a result of vitamin B deficiency, folate. So you could see how important nutrition is during pregnancy. So then, once these neurons are, once the neural tube is formed, then you have neurons and glial cells are born out, inside, and they start to immediately, as they are born, they start to migrate. They proliferate immediately, very, very quickly. About 250,000 neurons are born every minute. So because you want to have about close to 200 billion, so you continue producing them really, really rapidly, and then so the process of cell division that gives rise to new neurons is called neurogenesis. And then neurogenesis, as the neurons are born in huge numbers, they spread, they start to proliferate. And then we have kind of migration and cell differentiation. The old research tends to kind of look at differentiation as something that occurs after, but recent research, brain research, shows that differentiation is way, way early. It actually determines the order of migration. So you kind of look at it this way. You want to build a house, and you are making different pieces and, cutting, different pieces of wood and preparing them and so on. Does it mean, now, you want to build, say, the foundation of the house. Does it make sense that you start to manufacture things that are needed way later in the process before you manufacture the pieces that you will use for the foundation? So that is the rationale. That the fact is that cell differentiation, the cells that as the neural cells that specialize in the function that they are made to perform. Say for example, I want an eye neuron to serve to be a visual processing neuron. I want a neuron to serve as an auditory processing neuron, as a motor neuron, as a sensory neuron. So the original plan, the programmed genetic plan, is going to determine what kind of cell is needed, and when it is born, the ones that are specialized in a particular skills, particular cells that need to get a certain piece of the brain or the nervous system ready, they are born first. And when they are born first, they are specialized immediately. And then that specialization is going to, or differentiation is going to drive them to the site where they are going to stay and to function. So that is what the new research is showing us. You will find a lot of based on the previous kind of older research that speaks about differentiation as something happening a long time, before, after, but that is not the case. So the processes of neurogenesis, cell differentiation, and migration are very, very well synchronized. And all of them just are immediately happening at the almost at the same time. A cell is born, then it is differentiated, and it is specialized now, in whatever function it will do, to be, for example, a motor cell, an auditory cell, or whatever it is. And then it starts to migrate immediately as soon as it is born. Migration takes three different forms. One way is that some of the cells that are, inside the neural tube, first you need to build structures that are close inside of the neural tube, like the hippocampus, the hypothalamus. These are all pieces, parts of the brainstem. These need to be there, first, quickly. So they kind of develop right near, and the cells, as they grow, they push each other away. And, a cell is born, and then a new one comes in and pushes it away, and the new one comes in and pushes it away. So that is free cell migration. And it builds areas that are close, that are primitive, that are needed to make the new body that is forming, to make it kind of do basic functions. Another kind of migration is using axon tracks. There are some axons that have long tracks like threads, and some of the neurons grow and kind of are guided by these tracks. And then the neuroglia, these glial cells that form these tubes, and they guide, the radial glia guide neurons as they travel. So this is what is, in terms of the processes of neurogenesis, proliferation, differentiation, and migration. So differentiation is approximately, starts approximately day 25. And then neurogenesis itself, the birth of neurons and so on, is basically, it goes up to about day 108. And then now you have produced just all these neurons that you need, and then you need to connect them and to kind of, establish networks with them and so on. When after the brain is populated, then you have the stage of folding. The cerebral cortex, it continues to grow bigger and so on, and then because of the skull being limited in space, then you have the folding of the cerebral cortex into gyri and sulci, so that you can pack a greater surface structure. If the cell migration process gets disrupted, for example, in a certain kind of disorder called lissencephaly, you have clusters of cells, neurons, that are destined for the cerebral cortex, the very, very top layer of the brain. As they continue, something happens and they stop migrating, they get trapped inside of the white matter, beneath the gray matter, beneath the cerebral cortex. And they make clusters and they just, sit there. In one way, they disrupt the normal function of the white matter, because they are taking space that they are not supposed to take. And in another way, they are depriving the site where they are needed. That site doesn't have them. So that condition, lissencephaly, it results in in kind of a disorganization of the various layers of the cortex. The cerebral cortex, that little top layer that I mentioned in the previous class, that it is about 4 millimeters thick, it has 6 layers. And now in lissencephaly, it will have about 4. And in addition, because there aren't as many cells due to the lack of migration, that is going to cause a smooth and flat brain that doesn't have these deep valleys and prominent gyri. So lissencephaly. Cell maturation. Now, the cells, let's say, after the migration process is completed, the cells are in their destination now, and now you need to connect them so that they can communicate, they can connect with the different parts of the brain and with the different parts of the body eventually. So, but, in order to do that, they have to develop to be bigger, to have kind of more developed structures. So one of the first processes that newly or new cells that have reached their destination do is to grow new dendria, new dendrites on their cell bodies. And this is called arborization. Arbor means tree. Arborization is the process of growing branches. So a cell that is like this initially is going to grow rich branches, and each one of these branches has numerous sites for other neurons to connect with. So it has a rich and thick and healthy number of synaptic dendria or dendrites. Then, at the same time, then you have synaptogenesis. Cells are going to identify other cells that are specialized likewise, and they are going to connect with them. Usually, when cells fire at the same time, they know they are able to identify each other and they are going to connect. This is why the saying goes, cells that fire together wire together. So even before the environment gives stimulation, sensory stimulation, even before that, these neurons, as they want to identify other neurons and to grow, and they fire. As they fire, they are guided by special molecules in them that are able to identify not only the target neuron that they are going to connect on, but the exact spot that is most appropriate for them to connect on. So synaptogenesis is the formation of new synapses with other neurons. Then now you have connected local areas together and the neurons are connecting with nearby neurons. You need to expand your connections. You need to have a little town connect with a town outside of it or nearby it. And you need to have this connecting to distant towns and different countries even. So you need to have networks. Network formation begins and is present and obvious during the 3rd trimester. And it reaches adult-like kind of form near the 40th week of gestation, right before birth. But if a 6 months fetus is born, that fetus has the number of brain cells needed, and the fetus can survive. It's just a matter of maturation and making connecting these networks. The networks that the embryo they are genetically determined. However, they are on rough draft. They can be modified. And they are modified through changes in the environment. When the baby is born, the baby has less than 1/3 of the size of and the weight of the brain of the adult. The brain is about 3 pounds, 3.1 or so average. And the newborn is born with 0.9 of a pound. That's the normal weight. Of course, a mother who smokes is going to have a baby who has less, a brain that has much less weight than that. And it says on the packet, it leads to decreased low birth weight. And the low birth weight, in that case, it is not caused by, it is caused by slow maturation of the brain itself. So you do not have enough structures, you do not have enough connections, you do not have enough cells, to go and do the work that they are supposed to do, and so on. The process is that you come into life with these basic networks that are enough to run that little body, to make it swallow, to make it cry, to make it kind of move, to make it digest, and the basic functions that a baby a newborn can perform. And the fact that we have 66, 2/3 of the brain, grow outside of the womb, that means that the environment is going to shape that. And that most of that development of the 66% and the weight and, the increase and all of this, most of it is not by adding new cells. No, it is by making more connections. And as one of these videos that you have watched, it tells you, you make millions and millions of connections per second, every single second. So the brain has trillions of connections. So the environment is going to shape and stimulate that nervous system, including the brain. And depending on the nature of the stimulation or lack of that stimulation, the networks will develop and they will connect, and the brain is going to connect based on whatever the environment feeds into it, whether positive or negative. Every hour of the newborn's life, every single hour is absolutely critical for its development. Just think about how long a 3-year-old infant, how long does that child stay awake? How many hours of the day? You notice most, like 21 hours sleeping. And then the baby is available for you to socialize and to feed and to kind of for 3 hours. And then as the baby develops more and more and the nervous system develops more and more, then the baby is able to sleep less and to be free for that sensory stimulation for a longer time. But a day in the life of a newborn, a day that is lost without stimulation is absolutely devastating. And this is what I hope that we can find ways to enable parents and caregivers and anyone who works with babies to understand the critical importance of sensory stimulation by having babies see things, feel things, hear things, and interact and all of this. This is what the early years and early months of life form someone's life. And there are critical periods of development that we'll speak about in the next class that will show that for every particular period, there are stimuli and different experiences that must be provided in order for the system to work efficiently. So once these networks are formed and all these neurons that are not used, they are going to be kind of discarded. We'll see how this process goes, and that is part of maturation of the neuron. First, as I mentioned before, the neurons or the weight of the brain at birth is 1/3, a little bit less than 1/3 of the adult brain. And that would make you think maybe babies have less brain tissue, or they have less neurons than adults, but that is not the case. Newborns come to life with twice as many neurons as adults. And the estimates go from about 200 billion, and they go into about 100 billion or 85 billion that we have in the adult brain. And of course, somehow, a lot of these calculations, they don't take into account the cerebellum. The cerebellum itself has as many neurons as the brain, but they're condensed. So anyways, whatever the number is, the newborn comes to life with twice as many neurons as the adult. When you think of this, it just becomes kind of clear that when you think about the number of neurons in a particular area and how everything is jammed and everything is so crowded, you would not be able to have neurons that have room to extend up and down so that they connect with, deeper structures. Neurons that are going to be, or pathways that will form from back to front or sideways and so on. So the neuron itself, even though it is there, it is very small. It cannot do as much as an adult neuron does. So the idea is that this building of the networks is going to use all these, most of these neurons, and it is going to also make connections, a lot of connections. But you need to create space. You need to make these neurons that you are going to use, that are functioning now as part of the networks. You want to make them kind of bigger and more efficient, and they make more connections. As a result, as soon as the process of network formation begins, not far from, after birth, then you have the process of synaptic pruning and apoptosis, or spontaneous cell death of the neurons that are not used. So you have, for example, synaptic branches that are staying there and they're not connecting with anything else. These get, cut, they are disassembled, because like a tree, if you want the tree to grow taller, you need to cut the side branches. If you want the tree to grow, to become bushy and to spread around, you need to cut the top branches. So just exactly the same principle that allows the brain to focus its energy into and to invest it into things that are working and to make them work efficiently. And after that, all the stuff that is not being used, or all these synaptic connections that are made, sometimes that are not effective, sometimes they are not used frequently. For example, let's say that you meet someone, or you learn a word at some point, and then you haven't used that word in a long time, just, for a year, 2, 5 years, you do not hear about the person or the word, and then your brain figures out, that connection could be used for something else. This connection hasn't been used for a long time, and what is the use of holding on to it? So it's going to retract the synaptic terminal from the dendrite, and they will retract, and then that synapse is no longer there. So it gets severed and disassembled. So synapses that are ineffective, that are not connecting to the right target, synapses that are not being used frequently, all of these will be, and synapses that are not, and synaptic branches, dendritic branches that are not used at all. These are all disassembled. Now you create a lot of space around for, other things. But more importantly, whole, like a great number of these neurons themselves that are not being part of the network, they are not being used, they are going to undergo apoptosis, or spontaneous cell death that is programmed. So once they die, they create room around them, and the network then becomes bigger, and the other neurons that are still existing, they are actually protecting during that process. There are mechanisms to protect and optimize their function. I explain this process like this. If you get like a handful of corn, like some corn, and you just go and just spread it randomly and make it white condensed in an area, and you wait and see when the plants start to grow. If these plants, the corn plants, grow very close to each other, you will never have corn that you can eat. That doesn't happen. In order for make the corn plant, in order to make it grow, you have to create room around it, from all sides in a circle. That is going to give it a chance to grow bigger and to go up, to be taller and to have stronger roots. And this is how you can have corn. Look at any corn field. You have so much room around the corn plant itself. But if you want to have feed for animals, say at some point in the season, in some countries, that's a big source of just animal feed. They plant corn they make it very thick, and as it grows, it gets the plant, but it's very thin and tall, and it doesn't have it cannot develop to give you any corn. So again, the process of apoptosis and synaptic pruning are part of cell maturation. They also enable the existing cells to grow bigger and to make more connections, and the networks themselves to develop and to be more to function. It is not the number of neurons in the end. It is more of the connections that you make with the different areas that are nearby and the distant areas in the brain. That is what makes the biggest difference. Now, when synaptic pruning and synaptic and apoptosis doesn't happen, this process continues gradually until puberty. In puberty, there's a lot of reconstruction again, and a lot of networks are being refined, and the brain is highly flexible at that time around the puberty. But after that, it starts to have less flexibility, less plasticity. So that process of synaptic pruning and apoptosis, they continue going until you hit puberty. And one population that is known to have abnormalities in this area, in the synaptic pruning and apoptosis, is autism. Many studies show that this process doesn't progress, and in many cases it is decreased to the extent that a certain percentage of people with autism have more cells inside of their brain than the typical, and that means you have more, then you don't have room for the other ones to grow, and that means there's more local connectivity within an area, but you don't have connectivity across different areas, and you have difficulty integrating various senses so that you can kind of, in an effective way, so that you can get a coherent response, or that you can process things in a coherent way. So all of these things, we are speaking about very, very early stages in development, and yet you could see their effect, in many cases throughout life. Brain development follows an asymmetric pattern. Again, you need to think about, say you would like to build a house. What do you need to build first? So that asymmetry is driven by the functions that you need, that new body that is forming, to perform. You want that brain to make it perform these functions. What are these functions? You want immediately, as soon as you have, a brain, you want it to have control of the heart, you want to have control of lungs and different, structures and hands and all of this. So it makes sense that you develop these centers before the other centers, that are needed for other areas to develop. So the brainstem and the motor cortex, the brainstem is the oldest piece of the entire nervous system. As you see around that area, this is where the groove fuses, and then behind it and in front of it, then you have, the fusion continues from there. So the brainstem has functions like swallowing, breathing, heart rate regulation, blood vessel dilation and constriction, and stomach, digestion, different functions that are autonomic in nature, and they also reflexes. So also you have the motor cortex. These two areas experience a significant, accelerated growth prenatally before the baby is born. And after birth, for example, you have more of a faster growth in the cerebellum and other areas. By week 8 of gestation, after conception, the baby is able to feel touch. It has sensation on its the skin and various parts of the body, but by week 14, all the body is now covered with sensory extensions that are able to, to make the baby respond to touch. By week 20, the middle ear and the inner ear are adult size, and they are able to function, they are able to hear sound, and from that point in time, the baby is able to hear the mother's voice. The baby can recognize the mother's voice before birth, when they have a tape recorder and they tape the mother's voice and another person's voice, and they play them, and the baby is going to have an accelerated heartbeat when it listens, when the fetus, when it listens to the mother's voice. The same for native language of the mother. The baby is going to become more familiar with it and will be able to recognize it before birth and as soon as the baby is born. So the idea that when you have mothers, pregnant women reading in the last trimester to their babies, that is not a myth. It definitely is going to produce, to make the baby familiar with the language and with the sounds and with the rhythm of the sentences. The principle of brain learning depends on patterns. You hear every time you hear something, it will cause firing in the nervous system. first in the auditory system, and then when it goes to the cortex, it causes series and, of firing. And for example, when we hear a particular sound, that sound wave, it results from a number of vibrations. Let's say 100, whatever, based on your voice or whatever, a number of vibrations, and in the air, and then in the eardrum, and then inside of the cochlea. These vibrations are repeated exactly in every step of the way. Let's say that you hear a sound that is caused by a bird, and that sound has 500 vibrations, a high tone. By the time that 500 vibrations, by the time they go to the inner ear, they cause the hair cells to fire 500 times. So in 1 second. So that frequency of that sound is 500 per second, and then the auditory nerve is going to be firing 500 times per second in response to that. There are sounds that require, say, 200 times, that require 100, and different, depending on the signals that are received, the brain will fire a number of times that are identical, that is identical to the signals that are coming in. And as a result, of course, also it will fire based on the pattern. So if you do say fire, fire, and pause, fire, fire, and pause, then that's a pattern. And this is exactly how we learn words, how we learn sounds, how we learn the syntax of sentences. So you induce a neuron to fire in a certain pattern. It remembers that pattern. It becomes familiar, and then it masters that pattern. And then it will fire effectively and spontaneously without having to exert so much effort. Any kind of learning that we acquire is based on that, based on experience, based on continuous stimulation. So during the last trimester of pregnancy, the baby is able to get a lot of exposure to a lot of sounds, and that is going to enhance the baby's ability to learn these things after the baby is born. Taste also, research shows that, if you feed the mother in the last trimester a certain kind of diet, say carrots, more than others, and then the newborn, when they start to eat, then you see that they have a preference for carrots, or whatever the mother consumed more of. So taste buds at that time, they develop, and the baby in the last trimester has the ability to taste and to smell. Also by week 26, the baby has the ability to feel pain in different parts of the body. And so in terms of linguistic skills, language skills, if a mother reads, say, a certain story, and while she's pregnant and in the last trimester, research shows that the baby has preference for these stories that are read during pregnancy. And of course, you can now understand how you can shape the taste of the baby, how you can shape the baby's preferences for things, whether for stories you read with the baby, or for, say, you could read with them books about science, animals, and so on, or mathematics. So you can really take any skill at that time from that level and kind of go from little things to bigger things and make a specialist out of a baby. And this is, this is again based on the principle of how the brain learns and develops. So the effect of epigenetics on brain development is quite remarkable. And the interaction, the conditions that are in the family, the conditions that are present in the interaction that the baby has with the society, with the, with the parents and the siblings and the and so on. All of that is going to induce changes in the DNA of the baby, of the brain, so that is going to, to enhance the ability of certain genes to be expressed or to inhibit certain genes. Give you some examples. Say in animals, for example, when including humans, right after birth, for a period of time during the first few months, the baby must be exposed to light. And if there's deprivation, if one eye is sealed, for example, or one eye has cataracts, and it is left for 6 months, that is going to it's going to make the baby blind for life. Even if the eye was normal and it is sealed, or if there's, the baby lives in darkness all the time, that is going to deprive the child, will make the child blind, because there are rods and cones inside of the retina, and these do not connect unless you expose them to photons of light. And we are going to speak more about this, discuss this more when we speak about the environment and how sensory stimulation induces connectivity of the brain and makes the brain connect and form networks. The process of epigenetics, the science of epigenetics, studies how the environmental external factors influence gene expression, and how, for example, different factors affect development of the body and development of the nervous system and so on. So the major epigenetic factors that influence development in this case include nutrition. For example, if the mother is taking drugs and neurotoxins during pregnancy, that is going to have an impact on the development of the nervous system and the brain. Heavy metals like lead and mercury, they are known to cause damage to the nervous system, and especially at certain levels, they could really produce significant disabilities. Nicotine and alcohol. When the mother is smoking herself, of course, the baby's nervous system will grow at a slower pace, and that is going to cause a smaller weight and size of the brain. That is also going to cause alterations in the structure of the nervous system itself. And nicotine exposure is linked to hyperactivity and to inattention in children, whether before birth or after birth. Also, the alcohol. So this is if the mother is smoking. Secondhand smoke exposure. If the mother is not smoking, but someone is in the house who is smoking, or they're at work and she is exposed to a lot of smoke inhalation, nicotine exposure while she is pregnant, that is going to also have a devastating impact. Just think about it, especially living in areas like colder areas of the US, for example. In the winter, we have windows, doors are closed, and imagine you are living in an apartment or some kind of condominium. Windows are closed and doors, and you are recycling the same air. Imagine if there's someone who's smoking there. It doesn't matter who it is, whoever is living in that place is going to be inhaling that recycled air and constantly, day and night, breathing the nicotine. In the summer, you have air conditioning. Now windows again are closed and doors are closed, and you are doing the same. So there's a little gap in between winter and summer, like 3 or 4 weeks where you do not use air conditioning or heat, but most of the year now the exposure would be there. So these are really, really critical things, and we'll speak more about them. But from the prenatal perspective, alcohol is going to cause fetal alcohol syndrome, and that has its own characteristics, and depending on the level, it has a devastating impact on the baby's cognitive skills. Maternal stress and depression. The maternal stress and depression, especially stress, it causes elevation of the cortisol level in the blood, and that ensures that the blood pressure is high. It ensures that the stress response is elevated in the body. And if it continues for a long time, of course, it leads to heart disease. It leads to cardiovascular abnormalities, and that could lead to strokes or heart attacks and so on. The elevated stress response is of the mother, passes on to the baby, and the baby, when tested even 6 weeks after birth, the baby has that elevated stress response. And that, of course, leads to temperament abnormalities. The baby is easily agitated, restless, could be have hyperactivity, hard to soothe, and basically the rudimentary symptoms of attention hyperactivity disorder, and and then other things are going to develop out of that. So, and worse than that, research shows that that baby is likely to pass on these abnormalities to her own babies. So the second generation is affected. Maternal stress and depression. These symptoms that, causing the stress response, they are experienced by women who are pregnant, and who experience discrimination. We call it perceived discrimination. Perceived discrimination, it is called this way because the person feels that he or she is discriminated against. It doesn't matter if, the other person acknowledges it or not, but what matters is that the person feeling it is going to experience the effect of it. And research shows that in African American women, who experience discrimination at work or in the environment and so on, they tested the length of the chromosomes. Every chromosome has a cap on the four ends of it. How the chromosome is shaped. Each end is capped by a repeated sequence of amino acids, just repeated sequences to protect the DNA from degeneration. However, these sequences, these layers, they are called telomere, the telomeres. The length of the telomere goes by your age. Every year we lose a layer. Every year we lose a layer. So research consistently shows that African American women experiencing discrimination, they have shorter telomere length. And that means that they are aging at a faster, the aging process is accelerated, so that is going to make them live, at risk for living less because of discrimination they experience. And of course, if they experience it during pregnancy, that is going to pass on to the next generation, unfortunately. So the consequences are tremendous, and there's a lot that needs to be done in the society, in our schools, at universities, everywhere. There is so much that needs to be done, and people need to be aware of this information. So we are going to stop at this point, and then the next class we're going to speak about the effect of the environment and the effect of sensory stimulation through the various senses that we have on the connectivity of the brain and the nervous system. So thank you, and until we meet again, goodbye.